Somewhere in a hospital blood bank in sub-Saharan Africa, a transfusion technician is reaching for a specific bag of blood, not because of the patient's name, not because of their diagnosis, because of a single letter printed on the label. Oh, the most common blood type in the world, the one that emergency departments keep the most of in reserve, the one that soldiers have sewn into their uniforms, that disaster relief. Organizations stockpile before hurricanes that paramedics carry in trauma kits when there is no time to test the patient's own type. Type O. In a crisis, it saves everyone. What nobody told you when you found out what your blood type was is why. Not the clinical answer. The one about red cell, antigens, and antibody compatibility that your doctor could explain in 2 minutes. The deeper answer, the one written into the evolutionary history of this variant. The one that explains why blood type O is not just medically useful, but represents something remarkable about what human beings have survived. The one that required decades of genomic research, a series of discoveries about malaria and viruses and ancient DNA from people who lived 50,000 years before any hospital ever existed, to begin to understand. This is the story of what blood type O actually is, where it came from, what it protects you from, and why a single molecular difference in a gene on chromosome 9, a difference that looks, from one angle, like nothing more than a loss, has turned out to be one of the most consequential and most studied genetic variants in the entire human genome. Begin with what blood type actually is, because the clinical shorthand most people learn, AB, O plus or minus, obscures something genuinely interesting about the underlying biology. Your blood type is determined by the presence or absence of specific molecular structures on the surface O, F your red blood cells. These structures are carbohydrate chains, sugar molecules assembled in specific configurations and anchored to the cell membrane. And your immune system learns to recognize whatever is on your own cells as self, generating antibodies against whatever is not. If you are type A, your red cells carry A antigens, and your immune system generates anti-B antibodies. If you are type B, you carry B antigens and make anti-A antibodies. If you are type AB, you carry both antigens and make neither antibody. And if you are type O, you carry neither A nor B antigens. But your immune system generates both anti-A and anti-B antibodies, making your blood compatible with recipients of any type, because there is nothing on the surface of your cells for their immune system to recognize as foreign. This is why type O blood is used in emergencies. But the surface-level clinical explanation skips something fundamental. The reason type O red cells carry no A or B antigens is not that the gene encoding those antigens is absent. The gene is there. It is the same gene at the same location, the ABO locus on chromosome 9, that encodes the enzymes responsible for building A and B antigens in people with those blood types. In type O individuals, that gene carries a specific mutation, a deletion of a single nucleotide at position 261 of the coding sequence, that introduces what geneticists call a frame shift, shifting the reading frame of the entire subsequent protein sequence and producing a truncated, non-functional enzyme. The machinery for building A or B antigens is present in your cells. It simply does not work. Blood type O is, in the technical language of genetics, a loss-of-function mutation. This is not the intuitive picture most people carry when they think about their blood type. A loss of function sounds like a deficiency, something that should, if natural selection were operating cleanly, be weeded out of the population over time in favor of the functional versions. And yet, type O is not a rare or marginal variant. Globally, blood type O is the most common blood type. Approximately 44% of the world's population carries it. In some populations, particularly in parts of sub-Saharan Africa and among indigenous populations of the Americas, the frequency of type O climbs much higher, approaching or exceeding 80%. Whatever this loss-of-function mutation is doing, it has not been weeded out. It has, in substantial portions of the world, become the dominant form of the gene. And understanding why begins with one of the most powerful forces in human evolutionary history. Malaria, caused by the Plasmodium falciparum parasite and transmitted by Anopheles mosquitoes, has killed more human beings over the course of history than any other infectious disease. It remains one of the leading causes of child mortality in sub-Saharan Africa today. And for the approximately 10,000 years since the transition to settled agriculture, dramatically expanded the habitat available to the Anopheles mosquito, creating for the first time stable human populations living in close proximity to standing water and mosquito breeding sites. Malaria has been one of the most sustained and intense selective pressures acting on the human genome. Many of the most extensively studied genetic variants in human populations, sickle cell trait, glucose 6-phosphate dehydrogenase deficiency, variants in the hemoglobin genes, are present at elevated frequency in malaria endemic regions, specifically because they confer some degree of protection against the most lethal forms of the disease, even at the cost of associated health trade-offs under other conditions. The connection between blood type O and malaria became clear through a specific and somewhat counterintuitive biological mechanism. When Plasmodium falciparum infects a human red blood cell, it does not simply sit inside it passively. The parasite actively modifies the surface of the infected cell, inserting its own proteins into the cell membrane, proteins that then interact with the surfaces of surrounding uninfected red blood cells, causing them to clump together in clusters called rosettes. This rosetting behavior, infected cells binding to uninfected cells and pulling them together, is not a side effect of infection. It is a virulence mechanism. Rosettes impair blood flow through the small capillaries of the brain and vital organs, contributing to the most severe and fatal forms of malaria: cerebral malaria, which kills children through brain swelling and seizures, and severe anemia from the mass destruction of red blood cells caught in the rosetting process. The key observation, first documented in laboratory studies and then confirmed in field research with affected children, is that the parasite's surface proteins bind preferentially to A and B antigens on the surface of uninfected red blood cells when forming these rosettes. Type O blood cells, carrying neither A nor B antigen, are dramatically less susceptible to incorporation into rosettes. The infected cell's machinery for clumping cannot bind as efficiently to a cell that does not display the molecular targets it evolved to use. The rosettes that do form in type O blood are smaller, less stable, and less capable of causing the vascular obstruction that drives severe disease. The clinical evidence for this protective effect, published in the Proceedings of the National Academy of Sciences in 2007 by a research team working with children in Mali, was striking. In a matched case control study of 567 million children, blood type O was present in only 21% of severe malaria cases. The children who developed cerebral malaria and life-threatening complications compared with 44 to 45% of controls who had uncomplicated malarias or no disease at all. The statistical analysis showed that blood type O was associated with a 66% reduction in the odds of developing severe malaria compared with non-O blood groups. This is a large effect. It is not a marginal statistical association, but a difference substantial enough to be directly visible in the distribution of blood types between severe cases and healthy controls. The implication of this finding for the global distribution of blood types is direct and measurable. If blood type O confers substantial protection against severe, fatal malaria, and if populations living in highly malaria endemic environments have faced that selective pressure over thousands of generations, then natural selection should have increased the frequency of the O allele in those populations over time. And that is, with reasonable consistency, what the geographic distribution of ABO blood types shows. The highest frequencies of blood type O in the world are found in sub-Saharan Africa, where malaria has been endemic F for thousands of years, and among indigenous populations of the Americas, where some researchers have proposed that founder effects and other demographic processes contributed to extremely high O frequencies. European populations, which have experienced malaria pressure for a shorter period and at lower intensity, show lower O frequencies and higher A frequencies. The global map of ABO blood type distribution does not map perfectly onto the malaria map. There are other factors, including genetic drift, demographic history and selection, pressures from other pathogens. But the correlation is real and has been taken seriously by researchers studying both blood type evolution and malaria epidemiology. The loss-of-function mutation that produces blood type O was under the specific selective environment imposed by malaria, not a deficiency at all. It was a survival advantage. The enzyme that builds A and B antigens turns out, in a landscape where Plasmodium falciparum has spent millions of years, evolving to exploit those antigens as molecular hooks for its most lethal virulence mechanism, to be more dangerous to its carrier than its absence. Type O is not the ancestral version of the gene that type A descended from, as early models of ABO evolution sometimes suggested. It is a derived mutation, a modification of the original sequence, that, in certain environments, the human population has found advantageous enough to maintain at high frequency across thousands of generations. The ancient DNA research that has emerged over the last decade adds a further dimension to this picture. Researchers analyzing the ABO blood type of Neanderthals and Denisovans, using the same genomic sequencing technologies that identified those populations' contributions to the modern human genome, found that the type O allele was present in archaic human populations before the emergence of modern Homo sapiens. The Altai and Chagirskaya Neanderthals, sequenced from bone fragments recovered in Siberia, were both homozygous for O alleles. The Denisovan individual carried O alleles as well. Blood type O is not a recent innovation in the human lineage. It predates the separation of Neanderthals from the lineage that would eventually produce modern humans. It may be among the oldest blood type variants in the entire human genus, predating the emergence of our own species by hundreds of thousands of years. This does not mean blood type O is the ancestral state in a simple sense. The ABO locus has a complex evolutionary history with A, B, and O alleles arising and diversifying through multiple independent mutational events across primate evolution. And the relationship between them is not a simple linear descent from one ancestral type. But it does mean that the type O allele is old, that it has persisted across multiple human and archaic human populations over enormous spans of time, and that its persistence is not incidental. Something about carrying neither A nor B antigen on the surface of your red blood cells has, across the environmental pressures encountered by the genus Homo over hundreds of thousands of years, been worth keeping. The malaria story is the most extensively studied and best supported selective explanation for blood type O's high global frequency. But it is not the only one. The pandemic of 2020 produced a substantial body of research examining whether ABO blood type influenced susceptibility to SARS-CoV-2, and the results, though imperfect, and in some cases conflicting, showed a consistent enough pattern to attract serious scientific attention. Multiple large studies, including a population-based cohort study, OF 556 patients, published in the Annals of Internal Medicine, found that blood type O individuals had a measurably lower rate of SARS-CoV-2 infection and a lower rate of severe COVID-19 illness compared with non-O blood groups. Type A was consistently associated with elevated infection risk across multiple independent datasets. A systematic review and meta-analysis published in 2021 concluded that, despite between-study discrepancies, an overall consensus had emerged showing blood group O associated with lower COVID-19 risk. The mechanism proposed for this association is consistent with what we know about blood type O's anti-A and anti-B antibodies. All type O individuals, by definition, since they have neither antigen, produce both anti-A and anti-B antibodies as part of their normal immune complement. Researchers proposed that these antibodies may interfere with viral entry into cells by binding to A or B antigens on the viral envelope or on the ACE2 receptor that SARS-CoV-2 uses to enter lung cells, partially blocking the interaction. This remains a hypothesis rather than a fully confirmed mechanism. The COVID research on blood type moved quickly. Some studies were confounded by the demographic characteristics of the populations tested, and some well-designed studies found no significant association. The malaria connection is robustly established. The COVID connection is suggestive, but not definitive in the same way. What is notable is that the same property that protects against malaria, the absence of molecular targets that certain pathogens exploit, appears to confer at least partial protection against respiratory pathogens as well, suggesting that the absence of A and B antigens may function as a more general defensive configuration than any single disease-specific account implies. The third major clinical finding about blood type O concerns not infection resistance, but coagulation. Blood type O individuals have consistently been found to have lower plasma levels of von Willebrand factor, the protein that plays a central role in initiating clot formation at sites of vascular injury, compared with non-O individuals. The magnitude of this difference is approximately 25 to 30%, a substantial gap that has direct consequences for how blood clots form in response to injury or pathological conditions. People with blood type O are at measurably lower risk of venous thromboembolism, the formation of dangerous blood clots in deep veins, compared with non-O individuals, and this protection appears to operate through the von Willebrand factor pathway. Type A, B, and AB individuals have higher circulating levels of von Willebrand factor, and correspondingly higher rates of thrombotic events. This finding cuts in multiple directions simultaneously. Lower clotting risk is protective against deep vein thrombosis, pulmonary embolism, and the thrombotic complications that made COVID-19 so dangerous in severe cases, all conditions where excessive clot formation causes the pathology. But lower von Willebrand factor. Our levels also mean that type O individuals have a somewhat higher risk of bleeding complications in surgical contexts. And there is evidence of increased rates of peptic ulcer disease and other conditions where the clotting system's role in tissue repair is relevant. The same molecular architecture that reduces thrombotic risk introduces a different set of vulnerabilities. This is the standard evolutionary trade-off. A configuration that is advantageous in some contexts is disadvantageous in others, and what natural selection preserves reflects the net balance across all the relevant pressures rather than optimization for any single outcome. Taken together, the research picture that has emerged over the past 3 decades about blood type O is something considerably more specific and more interesting than the broad strokes of popular coverage have generally conveyed. The O allele is old, present in Neanderthals, probably maintained across hundreds of thousands of years of primate and archaic human history. Its high frequency in modern human populations is driven at least partly by a documented protective effect against severe malaria, mediated through a specific molecular mechanism involving parasite rosetting and the presence or absence of A and B antigens on red cell surfaces. The absence of those antigens also appears to interfere with some respiratory pathogens' entry mechanisms, producing a pattern of partial protection against certain viral infections that researchers are still working to characterize. And the lower von Willebrand factor levels associated with blood type O produce a measurably reduced risk of thrombotic disease at the cost of somewhat altered responses in contexts where robust clot formation is needed. None of this is the blood type personality profile that circulates in Japanese popular culture and social media array, where blood type is used to infer character traits the way astrology uses birth dates in other contexts. None of it is the blood type diet promoted by Peter D'Adamo, whose 1996 book claimed that people with different blood types should eat different foods based on their evolutionary heritage, a theory that has been studied directly by multiple research teams and consistently found to have no scientific support. When large epidemiological studies have examined whether individuals eating blood type compatible diets show better health outcomes in those eating incompatible ones, the answer has been no. The health associations with blood type O that the research has documented are with specific pathogens and specific coagulation mechanisms, not with dietary preferences or personality traits. What the research has documented is something both more specific and more profound than any of the popular accounts managed to convey. It is a story about a molecular loss, the deletion of a single nucleotide from a gene that became, under the specific environmental pressures of a world containing G. malaria parasites and certain respiratory viruses, a genuine biological advantage. It is a story about a variant that Neanderthals carried in the same caves where their firelit evenings left traces that modern genetics can now read, and that modern humans living in the malaria belt of sub-Saharan Africa carry at frequencies approaching 80%, because thousands of generations of selection have pushed it there. It is a story about how the same molecular property that makes emergency blood transfusions possible EEs, the absence of antigens that other immune systems would reject, turns out to be, in the evolutionary history that produced your body and everyone else's, a property that the human lineage has been quietly using to survive its most lethal parasitic enemies for longer than any written record covers. Your blood type O is not just a medical convenience. It is a document, a very old one, written in the language of molecular biology, whose full text we are only now beginning to read clearly enough to understand. Subscribe, because the smallest things in the genome turn out to have the longest histories.